Hello and welcome to week 2 of Biology 5404. Before I start the instructional part of this video, I want to tell you about something that I experienced in church last week. The musical group sang a little song called I Am Not My Own. I want to read some of the lyrics to you. The one who made the heavens made my heart and soul. Before I drew a breath, I was loved and known. A few more lines down, it says, my body is a temple of the living God. So as I heard that, I was thinking about this course, and it says, my heart and soul. We will be talking about the heart quite a bit in the cardiovascular system. Before I do a breath, we will be talking about the respiratory system in detail. We'll also be talking about all the other body systems like the nervous system, the digestive system, the excretory system. But all of those systems, when you put them together, still don't add up to be a human. Humans are more than just the sum of all of our body parts. And that, I think, compromises or makes up our soul. And so we're humans because God presented us with a soul. What I want to do now is share my screen with you and talk to you a little bit about membranes. Okay, let's see if this is going to work. All right, so here we see a piece of a plasma membrane. The blue parts are the phospholipids, the part that is impermeable to any kind of polar molecule. The colored things are proteins. This is an integral protein that extends all the way across. This is an integral protein that only goes part way. Here's another integral protein going all the way across, and we can see it's open all the way through. So this would be some kind of ion channel. Here's another integral protein, and this is what we would call a peripheral protein, which is just sitting on the surface of the membrane. By the way, this is intracellular, and this is extracellular. Here's another intrinsic or integral protein. The yellow things are carbohydrates. If a carbohydrate is attached to the lipid portion, it's called a glycolipid. If it's attached to the protein, it's called a glycoprotein. These proteins we see down here are parts of the cytoskeleton where the plasma membrane is attached to the cytoskeleton of the cell. So what we're going to look at today is how this membrane is involved in, first of all, creating a polarized situation for the cell, and how things can go into and out of the membrane using carriers and transport molecules, and how it sets up a charge distribution across the membrane, which can be used to do all kinds of work for the cell. So I'm going to the next slide now. Here we see a kind of protein which is called a channel protein. This is the channel protein right here. The characteristic of channel proteins is that they form a continuous opening between the extracellular fluid and the intracellular fluid. We have two kinds of channel proteins. We have some which are called open or leak channels. What that means is that they're nearly always open, and substances can pass through them by diffusion going down a concentration gradient. Then we have what are called gated channels. The channels have gates on them, and here we can see a little gate that is open. Here we see how the channel is closed. So we have four kinds of gated channels. They are called electrical channels, which means that the gate will open or close in response to a voltage change. We have chemical or ligand channels, meaning that the gate will open or close in response to a chemical. We have mechanical channels, in which maybe the gate is actually pulled open, or the gate may open or close when the channel is bent out of shape. And finally, we have thermal channels, which open or close in response to temperature changes. We also have carrier proteins, One of the main aspects of a carrier protein is it does not open all the way across the membrane at the same time. In other words, it's going to be shut on one side or the other. We have three classes of these carrier proteins. We have uniport, which carries just one substance. Like, for example, this carries only glucose. Then we have cotransport, and in cotransport, the molecules can be, two or more can be transported across the membrane in the same direction, in which is called symport, or they can be transported in the opposite direction, which is antiport. These transport molecules can be active, which means they're going to require some kind of energy to move the substance, or they can be passive, which means they can only move down a concentration gradient. Okay, here's the next slide. This shows that when a transport molecule attaches to its substrate, the substrate in some way or another is going to cause that protein to change conformation. So here we see how the protein is shaped this way, opening to the outside. When the substrate attaches to it, it changes its shape, and then it opens to the inside. So this carrier protein transports this substrate molecule from the outside of the cell to the inside of the cell by changing conformation after it attaches to the substrate. This can be active or passive. Here we see an example of a passive transport molecule. This is called GLUT, which stands for glucose transporter. This is an example of a protein that does facilitated diffusion. Glucose can only pass through this transporter down a concentration gradient, but the molecule facilitates that and makes it happen much, much faster than it would by having to go through the membrane. So here we see the glucose attaching to the protein here. That changes the structure of the protein, flips it open, and the glucose goes into the cell in this example here. But it could also do it in the opposite direction. If the concentration of glucose was more concentrated inside the cell than outside the cell, this transporter would move it in the opposite direction. Here we see an example of the sodium-potassium pump. You cannot overemphasize the importance of the sodium-potassium pump. Every living animal cell has functioning sodium-potassium pumps. And what the sodium-potassium pump does is to help keep a disequilibrium of charge across the cell's plasma membrane. This helps keep every plasma membrane polarized so that the outside of the cell is positive compared to the inside. So you can think of this charge distribution, this charge disequilibrium, as being like a battery that every cell has. And like batteries, this can be used to do work. So this protein is an example of antiport. In order to get it to work, where's my cursor? Here it is. 3 sodiums on the inside. First of all, let me back and remind you that as a general rule, there's about 12 or 13 times more sodium on the outside of the cell than there is on the inside. And we need to keep it that way to keep the cell's membrane polarized. But when 3 sodium ions on the inside of the cell attach to the protein, that will activate the ATPase, which is the enzyme part that breaks ATP. The ATP will be attached to here. It is split, and now we have only the phosphate left. That changes the conformation of the transporter, and it will open to the other side. Once that happens, the 3 sodium ions will be released to the outside of the cell. Now the protein is in a different conformation, and the potassiums, which are on the outside, will attach and be pumped on the inside. Now we have about 15 or 20 times more potassium on the inside of the cell than we do on the outside. But we have to keep it in there, so this potassium pumped in will pump it back in, release the potassium to the inside, and then this protein is ready to repeat that process. So the sodium potassium pump is continually running in all living animal cells just to keep the membranes polarized. One of the functions of the sodium potassium pump is to keep the concentration of sodium high on the outside of the cell. Now this is an example of co-transport. This molecule here is called SGLT, which stands for sodium glucose linked transporter. This molecule requires energy because we're going to be moving glucose from the outside of the cell to the inside, and it is more concentrated inside than outside, so the glucose cannot diffuse in. We have to pump it in. However, this transporter does not use ATP. It uses the energy in the high concentration of sodium on the outside. Remember, I likened it to a battery a few minutes ago. So as long as there's lots more sodium outside the cell than inside the cell, the sodium will try to diffuse in. When the sodium comes in and attaches here, that changes the conformation of the protein, and it can attach to glucose right here. So we see that in this thing, this figure. So now the glucose will attach. When the glucose attaches, that changes the conformation again, and the protein now opens to the inside of the cell, where both the sodium and the glucose will be released. The sodium will diffuse in because the sodium level is low on the inside compared to the outside. The glucose will come in because it has been transported. It can't go back out because this back end is closed. So we're moving glucose from a low concentration into a high concentration against the diffusion gradient. So this is how we can absorb glucose into our intestine, where we have a lot of glucose inside the cell, not as much glucose in the lumen, but we've still got to absorb it into the intestine to get it into the body. We also do this in our kidneys. This is actually a kidney slide. The concentration of glucose in our urinary filtrate is lower than the concentration on the inside of the cell. So down here, we have the sodium potassium pump, and remember, it's constantly pumping sodium out. As long as it is pumping sodium out, the sodium concentration inside the cell will be low. That means then that the sodium can attach to SGLT and then transport the glucose in. The glucose will come in, the sodium will come in, the sodium will get pumped back out, and the glucose will go through the facilitated transporter GLUT into the interstitial fluid, where it can then get into the blood. So this kind of transport of glucose requires direct active transport here, where the sodium is being pumped out using ATP, co-transport here, where the high concentration of sodium drives the transport, and diffusion here, facilitated diffusion through GLUT. So this is what a plasma membrane would look like. The inside of the cell is negatively charged compared to the outside, and we need that sodium potassium pump in order to maintain that charge separation. Now, you've probably heard about how the sodium potassium pump and the polarized membrane is responsible for action potentials in neurons and in muscles. We've also just seen how this high concentration of sodium is used to transport molecules by co-transport. I want to show you one other thing that this can do. Oh, first of all, here's another demonstration of the sodium-potassium pump. Sometimes it's called the sodium-potassium ATPase. By using ATP, it'll pump the potassium in, keep the potassium level very high in here, and it will pump the sodium out to keep the sodium level very high in here. The charge separation averages about minus 70 millivolts. Okay, now this polarized membrane can be used to do lots of kinds of work for the cell. I want to just show you one more. This is for the secretion of insulin. You probably know that the insulin comes from your pancreas, and that insulin stimulates cells to take up glucose, to lower the glucose levels in the blood. This is done by beta cells in the pancreas. So I want to orient you to all these molecules, and then we'll talk about how it works. This is GLUT. It's a glucose transporter that works by the facilitated diffusion. If the concentration of glucose in the extracellular fluid is very high, glucose will diffuse in rapidly. If the concentration of glucose out here is low, glucose diffuses in much, much slower. The glucose inside the cell can be used to make ATP. This is a potassium channel. As long as it's open, potassium will leak out of the cell to help maintain the proper concentration of potassium. This particular potassium channel is an ATP gate, which means if ATP attaches to this channel, the channel closes. It's an electrical gate. Here is the polarized membrane, and here is a calcium channel that is currently closed, and this channel is a voltage-gated or charge-gated channel, which means this channel will open if the membrane potential depolarizes. These are vesicles that contain insulin. The insulin is already in these vesicles. All it needs is for calcium to come into the cell to activate some proteins, which will cause these vesicles to dock with the membrane and release the insulin. So the way we see it now, we don't need any insulin being released because blood sugar level is low. Now, in the next slide, blood sugar level, blood glucose level is very high. So lots of glucose comes in through GLUT. This means that the metabolism of glucose is very high. Lots of ATP is made. So there is ATP available to shut the potassium channel. Now, when the potassium channel shuts, that means that the potassium cannot diffuse out. If the potassium cannot diffuse out, then it stays on the inside of the cell. Potassium is positively charged, so that makes the membrane potential go from minus 70 up to minus 60 or maybe minus 50. That depolarizes the plasma membrane. So this membrane here, instead of being minus 70 across it, is now only minus 50. The membrane has depolarized. That causes this calcium channel, which remember is a voltage channel, to open. That lets calcium come in. The calcium will interact with some proteins in the cell to cause these vesicles to dock and release the insulin. So, in summing this up, this particular process only works because the sodium-potassium pump keeps the membrane polarized. Now, I suggest that you come back and look at this video again after you have done all of the questions and videos and other exercises in this particular week. Okay, so that's all I have for this, and so I'm going to sign off.